Hello, my name is Maddie Shaklee, and I was working in the Fay Lab this summer. So my project is finding genome regions associated with pepper fruit shape using a genome-wide association study. So a little bit of background on pepper and its origins. So pepper is in the capsicum genus and is part of the Solanaceae family. There are 35 recognized species of pepper, and 5 of them have been domesticated. So my project specifically focused on C. baccatum. So we have C. baccatum var pendulum, which has been domesticated from C. baccatum var baccatum. So in this figure up here, this is the wild version, and in the bottom we have the domesticated fruits. So in the top, you can see that there are much fewer variety in fruit shape. They're all small and round. And then in the domesticated species, there's a much greater variety in shapes. So this pepper is almost exclusively grown in its native region in the Andes Mountains. So it's not something that we see commonly in this area. So then I want to figure out how can we identify the genes that are responsible for fruit shape variation just looking at the natural population. And that's where this genome-wide association comes in. So in order to do this, what we do is investigate the whole genomes of many individuals that have slightly different phenotypes in order to identify the genetic markers associated with the trait. So we do this by first identifying the single nucleotide polymorphisms that have significant genetic variation. So I have some examples of these in the gray boxes here. So the green SNP has both the wide fruit and the skinny fruit have the same allele, and they differ from the reference. And in the red SNP, the skinny fruit and the reference have the same allele, while the wide fruit has a different allele. So this is what the GWAS looks at for the entire genomes of many accessions and gives each of these SNPs a p-value based on how much association they have with the phenotype. And then from that, we can look at the linkage disequilibrium because the regions that have a highly associated are likely to contain genes responsible for the phenotype. So for this project, I looked at the pepper genome through computational studies to try to find the reason behind pepper fruit shape variation. And we want to do that to have a better understanding of the diversity and the genetic regulation for fruit shape, and to have better breeding and a better understanding of related species. So the hypothesis underlying this relies on linkage disequilibrium, which I mentioned earlier. This is the correlation between genomic regions that results in a non-random association. So if you have one and they are more likely to show up together, then it's going to be in linkage disequilibrium. And then it is possible to identify the genomic regions that are significantly associated with fruit shape from this genome-wide comparisons of SNPs with the phenotype data. So how I go about doing this is I start by calling the SNPs. So I have to find them in the genome, and then looking at the phylogeny and the population structure and genetic relatedness of the accessions. Then also looking at the phenotype data, the fruit shape variations, and then finally performing the GWAS. So starting off with the SNP calling, I have to map the Illumina data to the reference genome. So first have to trim the adapter and get rid of low quality reads. And then I can finally map the cleaned reads to the reference genome, which is shown in this part of the figure here. So the small, short Illumina sequences are lined up to where they match on the reference genome to figure out what genes they correspond with. And then once we've done this, we can mark the duplicates. I use GATK and then call variants. So once we've done this lineup of the Illumina to the reference, there are different types of genetic variants that can be found. So we call all of them, and then I just extracted the SNPs, which are the types of genetic variants that I was going to use. then moved on to SNP filtering, so getting rid of missing incomplete SNPs, and then also filtering for the minor allele frequency. I only wanted the most common alleles. And then did some SNP effect annotation. This would give me an idea of what each of the SNPs might do if they resulted in a change in the amino acid that was coded for, or might result in a premature stop codon, that sort of thing. And in the end, I got about 8 million SNPs to work with. So then have to look at the phylogeny and the population structure because the theory behind GWAS assumes that all of our SNPs are independent, but that is only true if all of our accessions are unrelated. So if we don't consider the genetic relatedness, we're going to get some false associations. So I started by looking at the phylogeny. I used SNPs at 4 DVT locations to generate this phylogenetic tree that you see here. I used IQ tree, and this is the maximum likelihood tree. So from this, you can see that there are some accessions that are more closely related to each other than they are to others. So that means that this will have a significant effect that we need to consider. So I also looked at the population structure. So I did a principal component analysis on a relationship matrix. So this relationship matrix compares every accession to every other accession and then gives them a value based on how related they are. Then doing the principal component analysis on that, I found there were 5 subgroups, which gave me these 5 subpopulations shown in different colors here. So this will come into play later when actually performing the GWAS. But before that, I have to look at the phenotype data a bit. So I have 66 total accessions. 2 of them are the wild type, and 64 are our domesticated accessions. So I have a wide range of fruit length data, and then the fruit width range was slightly narrower, and then also looking at the fruit shape index, which is the length divided by the width, it's between the two. I also had to do some data normalization because the raw data in these top histograms doesn't follow a normal distribution, so it's not going to work for the statistical tests later on. So I did a log2 transformation on all the data. You can see I get a much nicer normal data. And then we're ready for the GWAS finally. So this is a mixed linear model. So I did the MX, used the MX software, and I did it on the fruit length, width, and the ratio. Also with the kinship matrix and the principal component analysis that I talked about earlier, factoring in the genetic relatedness. I also had to calculate the number of effective SNPs to give me a threshold to figure out what should be counted as a significant SNP. How am I going to know if it's actually impacting the phenotype? So I got this red line from that on all of these plots. So these are Manhattan plots. So on the x-axis of all of these are the chromosome numbers, and on the y-axis are the negative log of the p-values that I got from running the mixed linear model. So from this, we can see that there's only any significant SNPs when we're looking at this bottom plot, which is the fruit shape index. So we found the most significantly associated SNPs on chromosome 10. So zooming in on that a little bit more, now we want to identify the candidate genes in this area using LD. So to start off, I selected the SNPs with the highest p-value, so the ones that are around this line or above, and then I calculated the LD for those using Plink, and then I wanted to take the SNPs with the highest LD. So LD is measured from 0 to 1, and I took the SNPs that have an LD of 0.8 and above. And then that gave me a region that I could check and try to identify what genes are in that area. So that gives me this area. This plot is zoomed in on that. And here, the SNPs in orange are the SNPs with the highest LD, and the purple SNPs have the lowest LD. So in the end, I found about 5,600 significant SNPs that have an LD of 0.8. And that gave me this region down here that is outlined by this candidate gene map, which has 18 candidate genes. So I looked at each of these candidate genes and tried to identify what kind of effect they might have by looking at what similar species have those sorts of genes. And I found this OFP2-like gene in teal here, which was very interesting. It is also almost directly below this triangle point here, which is the SNP with the highest p-value. So that makes it a good candidate. So this gene is the OFP, called an ovate family protein. So a little bit more about that. So OFP is found in tomato and in pepper. So OFP in tomato is called ovate, and it is known as a key regulator in tomato fruit shape. And it is mainly expressed in the reproductive organs, so mostly in the fruit. So here in this image, we have different tomato fruits that have different mutations in this ovate gene, and that results in different fruit shapes. So looking good. So there's also been found OFP in pepper through a linkage study of C. annum bar annum, which is another species of pepper, which gave the candidate gene OFP20, which encodes another ovate family protein. So now that we have a good idea of that protein, what are some of the steps that we would want to do next? So something to do would be to explore the effects of enhancing or silencing this OFP gene and see what that does to the fruit shape. We could also measure gene expression in different types of fruit shapes and seeing how much is expressed and at different points in fruit development. And then eventually this could have potential uses in breeding and hybridization to increase fruit shape. So in summary, I performed a GWAS to identify a candidate gene, a candidate region associated with the capsicum Bactem pepper fruit shape, and I found this OFP gene, which is a good candidate gene for fruit shape regulation. So with that, I want to thank my mentor, Zhanta Zhao, and my PI, Zhangcheng Fei, and then everyone in our lab, and also all of our funding for this project and for this internship.